noticed how the environment you're in, well, how much it affects you? Imagine trying to work in some cramped, rigid office versus, say, a spacious open one. Your stress levels, your ability to move, even how well you work. It all changes, right? Now, what if I told you that even down at the microscopic level, your cells are doing something similar? They're constantly checking out their physical surroundings, deciding how to live, how to act, based on whether they're like stuck on a flat surface or nestled deep inside a soft, complex 3D environment. Today, we're taking a deep dive into exactly that surprising idea. We're going to unpack a single, really fascinating research paper. Our goal is to pull out the core knowledge, the key insights, and basically give you a shortcut to being well-informed on this edge of cell biology. Our mission is to show how a cell's physical world doesn't just nudge it along, it profoundly dictates its health, its survival. We'll be focusing on some groundbreaking research published in the MBO journal. It's by FuiBoon Kai and colleagues. They look specifically at mammary epithelial cells, the cells in breast tissue. And they studied how these cells interact with their extracellular matrix, the ECM.
Right, the ECM. Think of it as the cell's neighborhood, the scaffolding and signals all around it.
Exactly. And the central mystery they tackled is pretty compelling. Why do cells, especially in these advanced models like patient-derived organoids, these are amazing 3D structures that mimic real tissue.
They're incredibly useful.
Right. Why do cells in these 3D setups behave so much more like actual tissue? Why are they more resilient to stress? Why do they secrete proteins correctly compared to the same cells just grown flat on a 2D lab dish? What's the hidden mechanism? What's the key difference driving this?
Okay, let's unpack this. So when we talk about cells behaving differently based on their environment, what's the core physical thing going on here? What did the researchers actually find?
Well, they zeroed in on something fundamental. It really comes down to a physical property called cortical actin tension.
Cortical actin tension.
Okay.
Yeah, basically you can think of it as the internal squeeze or the mechanical stiffness of the cell itself. Imagine the cell is like a tiny water-filled balloon. This tension is like the tautness of the balloon's skin. And it's controlled mainly by its internal scaffolding, the actin cytoskeleton. It gives the cell its structural integrity.
Okay, so this internal tension, this squeeze, it's influenced by the environment.
Yeah.
How does being flat, 2D versus 3D, change this tension inside the cell?
It's a really stark contrast. When these mammary cells are grown on a flat 2D surface, interacting with what's called a reconstituted basement membrane, or RBM.
That's the lab-made stuff mimicking the natural environment.
Exactly. In that 2D setup, the cells show high cortical actin tension. Their internal structure is pulled really taut, very constrained, like a tight drummed skin.
Okay, high tension in 2D, and the key difference in the more natural 3D setting.
That's where it gets interesting. When you embed the same cells within that RBM, so they're in a 3D arrangement, like in an organoid, they show significantly reduced levels of cortical actin tension.
So they relax a bit.
Precisely. The cell feels less constrained. It's like letting a bit of air out of that balloon. The skin becomes more pliable, more flexible. And this lower tension seems to allow for more, let's say, natural cell behavior. It's a big piece of the puzzle.
And you mentioned a specific protein involved in this tension control. Filamin. How does that fit in?
Filamin, yes. It's fascinating. It's an actin crosslinker. So its job is to help organize and stabilize that internal actin scaffolding we talked about. Now, in the high-tension 2D environment, the mechanical force actually causes filamin to physically unfold.
Unfold, like literally.
Literally unfolds. Think of it like a switch being flicked by the tension. This unfolding changes its shape, makes it highly active, and this active filament directly contributes to the cell's stiffness, keeping that tension high.
And in 3D?
In 3D, where the tension is lower, filament is less abundant at the cortex and seems more diffuse. It doesn't get forced into that unfolded, super active state as much, so it contributes to the lower tension environment.
Okay, so this internal tension is dramatically different. How does that physical state, that squeeze, then ripple through the cell's internal machinery? Where does it get really interesting inside?
Well, the effects are incredibly clear when you look at a critical organelle, the endoplasmic reticulum, or ER.
The ER, protein folding factory, right?
That's part of it, yes, but it's much more. It's this multifunctional hub, vital for proper protein folding, sure, but also calcium storage, regulating cell stress. Think of it as the cell's internal factory and its quality control center. Absolutely critical for keeping the cell and the tissue healthy, what we call homeostasis.
And what happens to this vital ER when the cell is under that high tension we see in 2D cultures?
The research showed pretty clearly that cells with high cortical actin tension suffer from significant ER dysfunction. It's a major problem.
Dysfunction.
They have issues with calcium homeostasis, basically lower calcium levels inside the ER, and they struggle to refill it. Calcium is like a key messenger in the cell, so that's bad news. They also show increased levels of specific molecular markers, signals that basically scream ER stress. So you get this overall ER stress phenotype.
And that filament protein, the one that unfolds under tension, it's involved here too.
Directly, that unfolding we talked about. It leads to filament interacting more with a key ER stress sensor, a protein called peak.
Ah, okay.
This interaction between the unfolded filament and P-arc basically sounds the alarm, activating this whole ER stress response pathway. It directly links the physical force to the biochemical stress signals.
Wow.
And what's really telling is that these high-tension 2D cells, they actually try to compensate. They increase connections that normally help refill calcium.
But it doesn't work.
It seems ineffective, almost like a desperate measure. Their calcium levels stay low, and the stress signals remain high.
So the big takeaway here seems to be, the physical squeeze directly impacts the cell's internal quality control. High tension stresses it out, makes the cell weaker.
That's a great way to put it, and the consequence. It directly reduces cell viability. These high-tension, ER-stressed cells become much more vulnerable to external challenges, things like chemotherapy drugs, paclitaxel, trail, doxorubicin, and even radiation.
That starts to connect the dots, maybe explaining why treatments might work differently depending on the tumor environment.
Exactly.
So what about the flip side? The cells in the low-tension 3D cultures, do they handle things better?
It's a night-and-day difference. Cells with low cortical actin tension, the ones happy in 3D, experience dramatically reduced ER stress.
So their ER is healthier.
Much healthier. They maintain good ER calcium levels, they have fewer active stress pathways, and they even boost the expression of genes that help with protein handling and protect against ER stress.
So reduced tension lets them function properly. And the practical outcome.
It translates directly to improved cell viability and, crucially, a robust resistance to those external stresses, chemo, radiation. It paints a clear picture. Physical tension inside the cell is a master regulator of its ability to handle stress and survive, and this is fundamentally linked to the health of the ER.
And this tension doesn't just mess with the inside, right? It also affects the cell's outer surface, the plasma membrane. How does it change the cell's skin?
It absolutely does. It sculpts its shape. The technical term is topology, and it affects the cell's ability to form protrusions.
Protrusions, like little fingers.
Exactly. Little fingers or bumps, the cell extends to feel its environment and, really importantly, to secrete substances, pushing things out.
So how does the tension level change these fingers?
The researchers used some pretty sophisticated computer modeling first. The models predicted that high tension would lead to short, fleeting protrusions like quick little jabs, whereas low tension would allow for longer, more stable protrusions like the cell is reaching out more persistently.
And does that hold up in real cells?
It did. They confirmed it experimentally. 2D cells with their high tension had these short, jumpy protrusions. 3D cells, low tension, had longer, more stable ones. And here's a cool part. When they artificially lowered the tension in the 2D cells,
It started acting like 3D cells.
bingo, they started forming those longer, stable protrusions. It showed the tension wasn't just correlated, it was causing the difference in membrane shape.
Makes sense. So if the cell's outer skin is shaped differently by tension, how does that connect to its ability to secrete proteins, getting stuff out?
Well, that change in membrane shape directly impacts secretion. High tension seems to hinder it. It might physically block the little protein packages of vesicles from getting to the surface. Or maybe it messes with recruiting the machinery needed to release them.
So it's harder to push things out when the surface is tight and dynamic.
That seems to be the idea. And conversely, in the low-tension 3D environments, they found more of certain helper proteins right at the plasma membrane, things like XS70 and ANXA2.
And these proteins help with secretion.
Yes, they're key parts of a machine called the exocyst complex. Think of it as the cell's docking machinery, guiding those secretory vesicles to the right spot on the membrane for release. So low tension lets the cell properly set up its launch pad for secretion.
It really is this intricate cascade, isn't it? High tension hinders secretion, which could then cause proteins to back up inside the ER.
Making the ER stress even worse.
Right. It's like a vicious cycle.
Yeah.
One problem feeding the next.
Absolutely. So another key insight here is The cell's internal squeeze doesn't just hit the internal factories like the ER, it fundamentally sculpts the outer membrane and controls secretion. Mechanics influence structure, structure influences function, it's all connected.
So let's talk about the so what.
Yeah.
What does this all mean for medicine? It sounds like this research gives a solid reason why those 3D organoid models are so good.
It really does. It provides a crucial mechanical explanation for why patient-derived organoids and spheroids work so well at mimicking real tissue.
Can you just spell that out? How does this tension discovery directly answer why 3D models are better?
It's their lower cortical actin tension. That's the key. Because the cells in 3D feel less squeezed, their ER functions properly good quality control. They can mature more naturally, secrete proteins the way they're supposed to, and they're much better at resisting stress. This makes them incredibly valuable, much more accurate tools for testing drugs, for modeling diseases. It finally gives us a powerful way to study human biology in a dish that actually reflects reality much better than flat cells.
And the implications for understanding diseases like cancer seem pretty significant too. You mentioned tumors often grow in stiff environments.
Right, a stiff, fibrotic ECM, much more rigid than normal tissue.
So how would this paper suggest that stiffness impacts the cancer cells?
Well, this research strongly suggests that such a stiff environment would crank up the cortical actin tension inside the cancer cells, just like we see in the 2D lab cultures. And that high tension could lead to chronic ER stress, changing how the cells behave, maybe making them more aggressive or perhaps more resistant to certain therapies. It's a clear mechanical link to how the disease might progress.
So the physical environment itself could be stressing out the cancer cells' ER, potentially influencing treatment response or tumor growth. What about genetic changes in cancer?
Good point. Many common cancer-causing genes, oncogenes, like activated RES, are actually known to increase this actin-myosin tension inside cells. So this research provides a bridge. It shows how those genetic mutations, by increasing tension, would also trigger this ER stress response signaling.
Wow, so it connects the genetic drivers of cancer directly to these mechanical stresses and the ER response.
Exactly. It adds a whole new physical dimension to understanding how cancer works.
Okay, so this study doesn't just explain things, it points towards new ways to treat diseases, maybe. You said the tension seems necessary and sufficient to control ER function and viability.
Correct. That finding is really powerful.
Does that mean we could target this tension? Develop drugs that, manipulate the squeeze?
That's the exciting therapeutic angle, yes. Because the actin cortex tension is so central, molecules that regulate its composition or mechanics become really attractive drug targets. We could potentially develop drugs to specifically loosen or perhaps tighten this internal tension.
How would that help?
Imagine a drug that reduces tension in cancer cells. It might alleviate their chronic ER stress, maybe improve how they handle proteins, and potentially make them more sensitive to existing chemotherapies. Or maybe in other diseases, you might want to increase tension if cells are too floppy. It opens up a totally different approach, focusing on the cell's physical architecture, not just its chemistry.
So this really raises a big question, doesn't it? Could understanding and maybe even tweaking these mechanical forces lead to entirely new disease treatments, moving beyond just chemical signaling to target the cell's physical structure?
It's an incredibly exciting prospect. I mean, this deep dive really highlights the profound role of cortical actin tension, this physical force in orchestrating so many fundamental processes: ER function, secretion, stress resilience, viability. It's a powerful reminder that mechanics, not just chemistry, are fundamental to how cells live and make decisions.
So the next time you think about your cells, maybe picture these hidden mechanical forces at work. This research shows us that the physical landscape of our bodies holds deep clues about our health. And it makes you wonder, what other biological processes, from how we develop to how we age, might be governed by these subtle mechanical cues? What else could be physically influenced that we haven't even thought to look for yet? It really opens up a fascinating new perspective on cell biology and medicine.